So hello everyone, and welcome to this, the second day of the EANA International Spring School on hydrothermal vents. This school is organized as a part of the 20th anniversary celebration of EANA, which is the European Astrobiology Network Association, and will seek to address hydrothermal vents, which, as we know, are prime targets for astrobiological research from numerous interdisciplinary scientific viewpoints. So after yesterday's journey through geochemistry, we move today to the diverse and wonderful microbiology of these fascinating systems. And today's seminar lecture is given by Dr. Michael Macey of the Astrobiology OU group. Michael obtained his PhD at the University of East Anglia, studying the impact of microbes on atmospheric trace gases, and he is now a postdoctoral research associate at the Open University, researching the diversity of microbial communities inhabiting extreme environments of astrobiological relevance. And indeed, in that vein, today's talk is entitled The Microbiology of Hydrothermal Vents and the Limits of Life, and we'll discuss some of the range of metabolisms and microbiological strategies that are in play in hydrothermal systems. Throughout the talk, please remember to post your questions for Michael in the chat box, and we will endeavor to answer as many of those as we can at the end of the session. So on that note, it is my pleasure to hand the virtual microphone over to Michael and say the stage is yours.
Okay, thank you very much. My computer reliably tells me that I am unmuted, so I will carry on in that manner. Thank you very much for the introduction, and thank you to the organizers for allowing me to give this talk and doing such a great job putting together the Spring School, especially during such troubling times. I'd also like to thank all of you for attending, whether that's live or watching this at a later date over the recording. So today, as was mentioned, we're going to be going over the microbiology of hydrothermal vents, but also using this unique and extreme environmental series in order to explore key metabolic processes and ecological and evolutionary principles that affect survival under extreme conditions. I'm going to click that. Okay, cool, that's now gone. There we go. So over the duration of this talk, I'll be reintroducing hydrothermal vents and then going over some key microbiology concepts. So for those of you with less of a biological background, this will hopefully serve as a useful primer to make navigating the later portions of the talk simpler. And for those of you with a biological background, this will hopefully be a nice refresher for some core taxonomic concepts. We'll then be going over the extremophilic microbiome of hydrothermal vents, so the resident microbes and how they are living in these extreme systems, the different types of hydrothermal vents and the environments therein, and we'll be touching on the role of mobile genetic elements, what they are and how they play a role in affecting survival in the vent systems. So, hydrothermal vents. As seawater circulates through the ocean crust, and mostly along the mid-ocean ridge, but also in other areas of tectonic activity, it is geologically heated, and this results in hydrothermal venting, with this venting resulting in the release of hot and anoxic water. So the first step in producing media that contains no oxygen is to boil the water, with this incredible heating provided by the geological heating of the Earth. It then mixes with the cold and oxic deep ocean waters, and this mixing also has profound effects on the chemistry of the fluids themselves. So it's worth considering the differences between the hydrothermal vents and the ocean itself, and I thought that the most apt comparison would be between that of a desert and an oasis. Albeit the difference in levels of water between the two are very obvious, nature documentaries have kind of conditioned people to think that the ocean in general is an incredibly productive and active and abundant environment full of macro fauna and flora. However, the ocean is incredibly nutrient poor for vast swathes of its area, and many of the resident microbes and other species that live there depend on a boom and bust cycle of living. When nutrients transiently become available, they grow and are then predated upon or killed by viruses. Whereas the veritable oasis in the ocean is hydrothermal vents, which are a much more productive system as a result of these geologically heated waters. So as the waters are heated, reactions occur between the water and the rocks. And this enriches the fluids of hydrothermal vents with an array of important chemical species. As we heard in the first talk yesterday, metabolisms require an electron donor and an electron acceptor. And these are supplied by the fluids of the hydrothermal vents in abundance. And it's worth noting, because it will be coming up throughout the talk, that sulfur-based species are especially abundant within these fluid chemistries. Now, the enrichment of the fluids depends on the underlying geology of the system. As we heard yesterday as well, Darwin was at first principle a geologist, and there is no clearer example of geology controlling the resident biology of a system than in hydrothermal vents, a principle that holds here as well as in a field in the United Kingdom. It's also worth noting that the rapid change in temperature as the fluid mixes with the oceanic water results in the precipitation of minerals, with these then also providing an additional source of essential compounds for metabolic processes. So now let's take a step back and consider some key microbiology concepts. So of the 118 known elements, if we look at what's common across the entire diversity of life, and what's common to all cells, we in fact see that there is a subdiversity that is common to all living things. And that is carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur, or CHNOPS as it's shortened in talks typically. Now, it's not to say that the trace metals and other members of the periodic table are irrelevant to life. They in fact play major important roles in the functioning of proteins and other factors within the cell. And there are, in fact, now lanthanide-dependent enzyme systems that are known, something that was previously considered completely impossible in biology, that have been discovered in the last 15 years. But it is, in fact, simply that it's these key elements that make up the essential building blocks of life. So it's worth noting, for the purpose of this talk, when I'm discussing life, it's based on what is present within modern living systems, because it's incredibly possible that there were previously alternate systems that were then simply replaced. So life requires three key components as we understand it today: a membrane to enclose molecules inside, allowing the concentration of an internal chemistry in the protection of potentially more delicate molecules. It needs a system of information transfer and a system of generating energy. So for the first step, the membrane to enclose molecules inside, this is supplied by phospholipids. Now, if you look at the structure shown here, you can see the tail is two fatty acids, the saturated and the unsaturated with the kick. And then there is also the head, a glycerol bound to a phosphate. And it's the charge on the phosphate in combination with the fatty acids that provides this molecule with a duality that is essential to its function in living cells. The head is hydrophilic and can interact with water, and the tail is hydrophobic and cannot. Meaning if you combine phospholipids in the absence of biology with a fluid, you get the formation of these micelle type structures. So it can form these single layer lipid spheres with the heads facing out and interacting with the fluid and the tails facing in. And you can also end up with a lipid bilayer sphere. Again, in the absence of biology, you can immediately start to see how this would allow for the concentration of specific compounds. And it's this bilayer that is made up by the phospholipids that provides the membrane enclosing molecules inside of a cell. Then for the system of information transfer, all life on Earth uses DNA. And we have a picture here of a nucleotide, which is the building block of DNA. It has the phosphate on the left, the sugar, deoxyribose in the middle, and then the nitrogenous base, of which there are 4 in DNA, with an alternate 1 used in RNA. The phosphate and sugar react with other nucleotides, forming this phosphate-sugar backbone of the famous double helical structure that is so well known in DNA. And we also see the nitrogenous bases, which bind in a conserved manner through hydrogen bonds, with adenine always binding to thymine and guanine always binding to cytosine, with this ATCG making up the code that underpins all proteins that are used for subsequent roles in the cell, and is also the system of inheritance. And then lastly, a system of generating energy, with this predominantly being done by proteins within the cell, proteins which are made up of amino acids, such as that shown on the screen. Now, living organisms use around 20 of a total diversity of over 500 known amino acids, with this specification unknown for the reason why some would be selected over others, possibly due to economics or some possible unknown advantage of this subgroup out of the 500. But you can see on the left, there is an amine group, on the right, our carboxyl group, below a hydrogen, and to the top R, representing the variable side chain. When one amino acid binds to another from the amine group of one reacting with the carboxyl group of another, this forms a peptide bond. And as peptide chains formed by the peptide bonds grow, they start to interact and bend and orient and form structures, which eventually result in the formation of these types of structure shown here. So this is glucosidase, an enzyme that provides a role in metabolizing carbon within a cell. But this is just one example of an essential function provided by proteins within the cell, involving energy generation and metabolism of the key bioessential elements. And once you have these components, you need a system of carbon acquisition and energy generation. So terms that will be coming up throughout this talk are heterotrophic and autotrophic, relating to carbon acquisition. So we are heterotrophic organisms in that we need to consume an external exogenous supply of organic carbon, whereas autotrophs are organisms that convert inorganic carbon, such as carbon dioxide, to organic carbon. So a good example for this would be plants. There is in fact a major diversity of microbes that are also capable of this lifestyle of carbon acquisition. Then for energy generation, there is phototrophy, the generation of chemical energy from light. Again, plants being a good example of this. And then there is chemotrophy, a term that will be coming up a lot during this talk, the generation of chemical energy from chemical reactions. So for the purpose of this talk, we will be using and referring back to life, the definition that is currently used by NASA of a self-sustaining chemical system capable of Darwinian evolution, with the self-sustaining chemical system supplied by the membrane enclosing the chemicals, and then the proteins maintaining the internal cell state, and Darwinian evolution provided by the hereditary molecule of DNA that's passed between generations. Once you have life, and as all life on Earth shares a common origin and therefore a universal common ancestor, you can assess the relative evolutionary relatedness of different organisms using the type of diagram shown here, first drawn by Charles Darwin in Origin of the Species. So this is the first phylogenetic tree, showing the evolutionary potential relatedness of different species as he was sketching. And if we go to the utmost level of classification, the level of domain, we can, in fact, use this system. So all life on Earth can be classified into either bacteria, archaea, or eukaryote. Now let's open some of these branches up and have a look at what's present within each one. We can further combine bacteria and archaea into prokaryotes, which means that the DNA is loose within the cell, whereas in eukaryotes, it's contained within a smaller membrane-bound subunit. Bacteria and archaea, whilst all single-celled, vary from each other in several significant ways. So for example, bacteria, the phospholipids that make up their membrane shown at the bottom have the fatty acid tail that was introduced, whereas archaea have isoprenoid tails. Bacteria have ester linkages and right-handed glycerol molecules, whereas archaea have ether linkages and left-handed glycerol molecules, and both still have this key phosphate group. In addition to this, the regulation of gene expression is divergent between the two, with archaea having a much higher level of control over their gene expression. So if we go back to the combined tree, is it just that the DNA is contained that differentiates bacteria and archaea from eukaryotes? And overwhelmingly, no, it's not just the nucleus, which is membrane bound, there are in fact many membrane-bound subunits within the cell that perform assigned functions. These are known as organelles. So in addition to the nucleus, there is the mitochondria, which is the powerhouse of the cell involved in energy generation. And whilst we do not have them in our cells, there is also the chloroplast, which is involved in the harvesting of light energy, in addition to cell walls, which are also found in plant cells. Furthermore, to the membrane-bound organelles, we can see that this representative of prokaryotes here, we have an average range of prokaryotic cell sizes, although we now know that there are much smaller prokaryotes than this. But we can see that the largest prokaryotic cell is still dwarfed by the smallest eukaryotic. So this is another key difference, is the size of the cells, which then also impacts surface area to volume ratio. We go back to this tree once more, we can consider what is below the utmost level of domain. So this figure is useful for exploring some of the diversity within each of these classifications. So on the right in brown, we have us on the animal branch, next to the fungi, plants, slime molds, and amoebas. Then on the left, we have the bacteria in blue. The species of which people tend to be most familiar with are those that cause disease in humans, plants, or other animals, are beneficial to us in helping plants to grow, such as in the association with the leguminous plants, or produce important compounds. However, the overwhelming majority of bacteria have no interaction with us on such a personal level, but can in fact have a major impact on biogeochemical cycling of elements. And then there is the group with which people tend to be least familiar, the archaea. Now, we have some great examples of archaea here. We have the halophiles at the top of the red branch. These are the salt-loving organisms. And at the bottom of the archaea, we have the heat-loving organisms with thermo and pyro as the prefix of their name. Then in between this, we have the prefix methano, representing the methanogenic archaea, a metabolic process that is exclusive to the archaea, the biogenic production of methane in order to generate energy. However, whilst useful, the tree does promote an out-of-date view of archaea in that they are just extremophilic. And we now know that in fact, there are archaea present in every environment in which we find bacteria, and there are much more what we would consider normal archaea. Now, if we want a better understanding of the current understanding of the level of diversity of microbial life on Earth, we need to turn to Dr. Laura Hugg's seminal paper from 2016. Now, don't be overwhelmed by the levels of detail in this incredibly impressive figure. I will walk you through the key steps now. So this large bulb at the top here represents the total known diversity of bacteria, with the branches with red dots representing the clades for which there are no isolated representatives. These are only known through sequencing efforts. And then down at the bottom with this green tail, we again have us next to the amoebas. So this is the eukaryotes. And using these multicolored lines here, we have the archaea. Again, keeping their branching closer to the eukaryotes, with eukaryotes believed to have evolved from archaeal origin. So if we collapse these branches back down and go up to the level of domain, we can see that there are multiple levels at which we can classify organisms. And let's go over some of these now. So these are the levels of taxonomic classification. We've seen domain, and there are some of these that you may be familiar with. Genus and species tends to be most commonly used in research papers. There are also other levels and sub-levels, especially when organisms have a complicated taxonomic classification. But for the purpose of today's talk, we will be sticking to the more established classifications. And one which people are typically most familiar are us as a species. So this is an image from the Voyager probe, and we are eukaryotic organisms, we are animals, we have a vertebrate, we are mammals and primates. Now, this is how humans are classified using every level of taxonomic classification. However, if we look at, say, a dog, specifically my dog, Cosmo, with a picture taken here in the center of Milton Keynes, we can see that he is also eukaryotic, he's an animal, he has a vertebrate, and he is a mammal. And it's only when we get to the level of order that we can differentiate a human from a dog. Meaning that if you were to say what's in the next room, the answer and the level of information you were given would depend very much on the taxonomic level that was used. So for example, if it was just given to the level class, that's an incredible diversity of organisms with an incredible variation in ability and identity. Now, whilst flipping, this is still a useful example of the level of taxonomic classification, sometimes removing data, but sometimes providing excessive data with the level that we need to use depending on the question, the relative organisms that are present within a system, and what we're hoping to understand. So what if we look at not an animal? What if we look at a bacterium? Well, here we have E. coli, probably the most famous of the bacteria. In top, we have it in a plate, and in bottom, we have it under a microscope. So Escherichia is where we get the E from, and coli. So Escherichia coli. But there are levels below species. And if we had somebody come to you with a sample of E. coli and they said it was strain K12, it would presumably be from a lab or an industrial bioreactor, with strain K12 being an incredibly domesticated and friendly strain of E. coli that we use for the production of industrial and medical compounds. Whereas if somebody came to you with a sample of strain 0121, then this would presumably be from either sewage, a contaminated reservoir, or a very, very sick animal. So in order to compare between different E. coli samples, we need to go one level lower to strain. But what if we compare E. coli to a completely different microbe? Well, if we compare it to the most famous archaeon, Halobacterium, we can see immediately at the level of domain that it is different and distinguishable based on the utmost level of taxonomic classification. But we would only have an indication as to what these organisms were exactly and try to infer what they might be doing within the environment if we were to then look at lower taxonomic levels. Now, for the sake of this talk, as it is on the microbiology of hydrothermal vents, it's unfortunately not possible to completely get away from the Latin and taxonomic classifications. But what I can do is instead focus on simplifying this so that everybody can go through this talk together and it's not simply an overwhelming of names. Now, there are three usual suspects that typically come up in hydrothermal vent systems, and I will be using shortened names for all of these. So we have one of them shown here, another gamma proteobacteria like E. coli, but incredibly different if we get to the lower levels and what exactly they're doing in hydrothermal vents. And every time it comes up, I'll be using this color, and I'll be referring to it as gamma in the interests of shortening the long name. There is then this other organism, which is classified to a different phylum. This is a recent occurrence, and much like bands or singers, bacteria and archaea sometimes undergo changes of name. So every time this microbe comes up, I'll be calling it Campyllo. And if you've done some reading prior to this and you read about epsilon proteobacteria, that is this group who is recently reclassified. And then the third usual suspect we can see is again classified to a different phylum, and I will be referring to this as aqua because I also struggle with pronouncing this name. So I'll be using these three colors consistently throughout the talk as these organisms come up: gamma, Campyllo, and aqua. And any other organism that comes up, I will be using this green color, and I will go through specifically what the other organism may be doing as well. So why are the microbial communities of hydrothermal vents distinct from that of the ocean? Well, we've gone over that they're much more productive, but they're not just distinct from the general ocean community, they're also distinct from many other ecosystems on Earth. And that's because instead of being fueled by photosynthesis, they're fueled by chemosynthesis. Now, they're below the photic zone, but that doesn't mean that photosynthesis doesn't occur at all. The glow from hydrothermal vents has been shown to be able to support photosynthesis. But in the face of the abundance of reduced chemical species, chemosynthesis overwhelmingly dominates. They were first identified in 1977, and they were the first chemosynthesis-driven system that was ever known, and this has had a fundamental impact on several scientific fields, but no more than in microbiology. Since there have been other chemosynthesis-driven systems identified, such as sealed cave systems, such as Movila Cave in Romania, where again, there is the absence of light, so the ecosystem is driven by chemosynthesis. So the hydrothermal vents and the surrounding area as a result contain abundant reduced chemical species that can support a range of microbial metabolisms, but with some that typically come to dominate. And as previously mentioned, the fluid chemistry is dominated by the geology, but the chemistry of the region is also affected by the rate of mixing with the water and the rate of dilution with the seawater. In addition to the abundance of reduced chemical species that can be used to fuel metabolisms, there are many survival stresses that restrict the immediate environment of the hydrothermal vent to extreme fires. So these stresses include pH, pressure, temperature, and the high concentrations of metal ions and reduced chemical species that can fuel metabolism. Because there is, of course, no such thing as a safe chemical, there are safe concentrations, and they are present in such high abundances around the hydrothermal vent that these can prove toxic to many organisms. So what are extremophiles? As with many things in biology, extremophile is an anthropocentric relative term based on what we would classify as normal, but they are the organisms that both love extremes and can very often only survive in that exact extreme. These are microbes that are capable of surviving at the edge of a physical or chemical extreme. And whilst there are extremophilic animals and plants in multi-celled organisms, the most extreme extremophiles and the majority of extremophiles in general are single-celled. So walking through some of those, we have the thermophiles, and we're keeping up the image of one of the most famous extreme environments on Earth, the Grand Prismatic Spring from Yellowstone National Park in America. And you're a thermophile if you can survive above 45 degrees centigrade, and a hyperthermophile above 85 degrees centigrade, with the maximum temperatures in which cells can survive being above 120 degrees. There is often this delineation between standard version of the extremophile and extreme version of the extremophile for every category. And the issues that the cells face in this type of environment relate to the three things that cells need: a membrane, a system of energy generation given by proteins, and DNA. So the membrane can vibrate apart due to the higher levels of kinetic energy. The DNA is also damaged as a result of the higher levels of kinetic energy from the temperature. And proteins can denature, losing their folding and therefore losing their function, and no longer being able to maintain the internal chemistry of the cell. And there are many strategies to try and overcome this. So if your membrane is vibrating apart, you can change its composition with the addition of isobranch chain fatty acids, which help maintain the rigid structure. You can also increase the production of what's known as heat shock proteins. And these bind to other standard proteins in order to prevent them from denaturing, allowing their function to continue. And then there is also the production of DNA repair proteins, which try to repair damage inflicted on the DNA molecule of the cell. There are the piezophiles. So you're a piezophile if you can survive above 10 megapascals, and a hyperpiezophile above 50 megapascals. And the problems faced by piezophiles are very similar to thermophiles. They have issues with the membrane lysing, or in this case, also becoming impermeable to certain things being able to transport across. And proteins can also destabilize, once again losing function. The strategies here are very similar, but instead of increasing the concentration of isobranch fatty acids, they add unsaturated fatty acids in order to alter the membrane composition and make it more resistant to these higher pressures. It's also possible to reduce your rate of cell division, with this preventing the introduction of a weakness within the structure of the cell's membrane, and also accumulating osmolytes, such as glycine betaine, to help maintain the internal chemistry of the cell under this selective regime. Then there are the halophiles. So shown here, we have an image of Western Sahara, these salt flats in Western Sahara. And you're a halophile if you can grow above 0.2 molar sodium chloride, with extreme halophiles being above 2.5 molar sodium chloride. So just to put this into context, the average concentration of sodium chloride in seawater is 0.6 molar. So water is lost from the cell as a result of osmosis, and this results in the denaturing of proteins as well as the lysing of the cell. And there are two distinct types of strategy used by microbe to adapt to this. You can accumulate osmolytes within the cell, again, glycine betaine or ectoine or DMSP, in order to try and maintain the internal chemistry. You can try and constantly pump solutes in to balance the internal chemistry, including chloride ions and potassium ions. Or you can simply allow the salts to move into the cell and instead have proteins that are adapted to being in this osmotically altered state. And then the last group that we'll be stepping on today, the acidophiles. You're an acidophile if you can grow below pH 5 and a hyperacidophile below pH 3. And again, DNA can be damaged due to the loss of the internal chemical environment and the high concentration of hydrogen ions or protons, and proteins denature due to this loss of internal pH. Shown here, we have an example of a series of acidic hot springs that are studied by one of our research students in the group, Vincent René. And again, we see the expression of DNA repair proteins to try and minimize the damage to the DNA molecules. We see the expression of chaperones to try and maintain protein function. The membrane itself is altered in composition to prevent hydrogen ions so easily being transported in. And we see this constant energy expenditure to pump protons out of the cell and pump protons out of the cell and bring potassium ions in to try and maintain this internal chemistry. And as has been touched on, in hydrothermal vents, there isn't just one of these pressures, meaning that many of the organisms that occupy this environment are polyextremophiles. We can see in the archaea here, we have Thermococcus, a genus which contains hyperthermophiles, radiation-resistant microbes, pH extremes, as well as piezophilic microbes. And if we go over here to the bacteria, we see Geobacillus, which again contains thermophiles, halophiles, and microbes that are capable of surviving within pH extremes. So in no other case is there such a clear example of the fact that higher and lower evolved organisms and more evolved organisms being a false concept in terms of biology. Success of a strategy depends on selection, which depends on the environment. And these organisms have evolved a strategy that enables them to thrive in extreme environments and the most extreme environments on Earth. There is, of course, another way to enhance survival, which is widespread across the tree of life and not just restricted to extremophiles. And it's one that's employed by many hydrothermal vent-dwelling bacteria. It's biofilm formation. So this is a strategy that microbes use when they're exposed to an external stressor. This doesn't have to be an extreme stress. This could be something as simple as microbes in a pipe undergoing a current or exposure to antibiotics in a human. It requires communication between cells of the same species. So what happens is these cells are exposed to an external stressor, and they then attempt to adhere to a surface and produce this mucus-like substance. As they cannot produce enough of this on their own, they release communication molecules known as HSLs, and they communicate with other members of their species in order to more effectively coordinate the production of this mucus-like substance. Over time, this builds up and results in the formation of fully formed biofilms, with the mucus substance itself protecting some of the cells, but also the interior cells being protected by the exterior cells. This is something that's also observed in humans in tissues when they're exposed to antibiotics, but it's also a way of overcoming some of the extreme pressures within hydrothermal vents. It's also worth noting that some of the experiments that have been done on the ISS have noted that microbes that are capable of surviving exposure to space conditions use some type of assemblage similar to biofilms, with the external cells dying, but the internal cells of the same species being able to persist. So why do we research extreme environments? Well, there is genuine value in trying to answer the question, what are the limits of life on Earth? Understanding microbes that can inhabit extreme environments allows us to try and understand key evolutionary processes and processes in our evolution. With the theory that life went through a strong bottleneck very early on, and that LUCA, or most life that was evolved after LUCA, may be descended from extremophilic microbes. There's also, again, the anthropocentric point that many extremophiles produce novel compounds that have industrial applications, such as medicines and cosmetics, with extreme environments potentially representing an as of yet untapped reservoir of novel antibiotics. And then, of course, this being part of an astrobiology series of talks, there is value in trying to identify what are the strategies that enable viability at the most extreme points on Earth. What are the metabolisms and other survival implementations that allow these organisms to thrive under such extreme selection pressures? And can we use this to inform our understanding of potential habitability and viability on other celestial bodies, either current or under former potential environments and regimes on these celestial bodies, and the evidences that may be left as a result? So what are the different types of hydrothermal vents? It's not as simple as there are just deep sea hydrothermal vents, of course. There is a great diversity in terms of their setting, geology, and the rate of seafloor spreading. Now, broadly, using seafloor spreading as a criterion, we can categorize deep sea vents into 5 different categories, from ultra slow to super fast, with the spreading rate also having a major impact on the associated fluid chemistry. So for fast and super fast hydrothermal vents, these are associated with mafic rock and are commonly found along the mid-ocean ridge. And as mentioned, sulfur-rich species tend to dominate these fluids, specifically hydrogen sulfide, with sulfide being the most reduced form of sulfur. Whereas for these slow and ultra-slow hydrothermal vents, these are associated with mafic, but also ultramafic rock. And it's also found along the mid-ocean ridge, but also with core complexes. And these fluids are enriched not only in sulfur-rich species, but also methane. And then lastly, there is the other type of hydrothermal vents, which are associated with either felsic, dactic, or andesitic geologies. I'm sorry for butchering the names of the different geologies there. And these occur in association with volcanic arcs. These vents tend to be lower in pH, but richer in hydrogen ions, methane, and metal ions. And of course, it's not as simple as there is a hydrothermal vent and we can consider that as one environment. If we were to go to a forest, we wouldn't consider all plants as equivalent. And indeed, if we were to look at one plant, we wouldn't consider the roots as equivalent to the leaves. So instead, we're going to go step by step through each part of the environment. And first, we're going to start with the chimneys. So as water is emitted from the vents, it's rapidly cooled as it comes into contact with the seawater. As mentioned, this causes precipitation of minerals, and this results in the formation of chimneys with a black smoker shown here on the left and white smokers here on the right. The mineralogy of the vents, of course, depends on the geology as well as the rate of mixing. And this means that it can produce the black smokers or white smokers, which are sulfide and carbonate rich respectively, and can also result in the formation of various shapes with just chimneys shown here. Now, the immediate fluids are, of course, incredibly concentrated, hot, and anoxic. So in the immediacy of the mouth of the vent, this is, in fact, not colonized, with microbes only being able to start colonizing this environment once the temperature reaches genuinely habitable limits. And where this occurs, they are colonized first by hyperthermophilic and anaerobic microbes. It's not until further away from the mouth of the chimney that aerobic, so microbes that require oxygen, and aerotolerant microbes, so microbes that can survive in the presence of low concentrations of oxygen, can start to colonize. This requires greater distance from the mouth of the vent. And this steep gradient in temperature and reduced chemical species away from the mouth of the chimney as we go down an increased distance results in a strong gradation in the communities that develop along the chimney. So shown here, we have a representation of a mafic chimney, the fluids dominated by hydrogen sulfide. Of course, this wouldn't be as close as this due to the high temperature, but just for illustrative purposes. At the closest to the vent, they're dominated by methanogenic archaea. As previously mentioned, the group of methane-producing archaeal microbes, which contain some of the most hyperthermophilic microbes that are known. And at distance and in order, we typically observe the presence of aqua, campylо, and gamma, all of which are sulfur oxidizing bacteria. Now, let's go through these groups in more detail. Methanogenesis. Now, if I was giving this talk 5 years ago, I probably would have said they produce all biogenic methane. However, it's now known that there are some metabolic processes performed by bacteria, which as a waste byproduct, also produce low concentrations of methane, such as nitrogen fixation with iron-containing enzymes and reactions with methyl mercury. However, I believe it still holds true that the majority of biogenic methane is in fact produced by methanogenic archaea. Now, this is the hydrogenotrophic pathway, using hydrogen and carbon dioxide to produce methane and water. There are alternate pathways known that use acetates or one carbon compounds as either the electron donor or acceptor, but the below pathway that's shown here is the dominant pathway in hydrothermal vents. And this is a metabolism that often receives a lot of interest in terms of astrobiology and astrobiological questions because of the detection of methane in the atmosphere of Mars and its transient seasonal nature, which would imply that it's being replenished. But of course, this could also be geological in origin. Then there is the process of sulfur oxidation. Sulfide is the most reduced form of sulfur and is highly abundant in hydrothermal vents. Now, it can occur in the presence of oxygen, but it can also use alternate terminal electron acceptors, such as nitrate. Nitrate isn't incredibly enriched surrounding hydrothermal vents, but nitrate can be drawn in from the surrounding oceanic waters. And sulfur oxidation is also, in an anaerobic context, considered as a potentially relevant metabolism under Martian conditions, given that there is the abundance of sulfur species that have been either detected as present on the surface of Mars or in Martian meteoritic material, and the detection and reporting of nitrates as present within Martian mudstones in the Stern et al. 2015 paper. Major sulfur oxidizers in the hydrothermal vent systems are the Campilo, Gamma, and Aqua. It's worth noting that there are also sulfur oxidizing archaea. We won't be discussing them further in this talk because they're typically absent from the vents. So sulfur oxidation is an ancient metabolism, and this is reflected in the fact that it is in both the bacteria and the archaea. It's also incredibly widespread within those domains, and it's incredibly diverse across the tree of life. And this is an indication that it's an ancient metabolism. So we've heard that chemosynthesis fuels ecosystems in these environments without light. And what that means for many of these hydrothermal vents specifically is autotrophic sulfur oxidation, driving the primary production. This is the chemosynthetic pathway, with the capture of carbon dioxide by the autotrophs, then providing organic carbon for other members of the community. So in this manner, we step upon a key ecological concept, the concept of a keystone, such as the keystone in an arch, which allows the arch to stay up. In a beech forest, beech trees would be the keystone species, the species upon which the rest of the ecosystem depends. And by fueling primary production, the sulfur oxidizers could potentially be considered a keystone species. Or, given this is a metabolic process that's being performed by representatives of different phyla, a keystone metabolism, with the exact taxa being irrelevant to this process. However, we need to consider if there's these three microbes that are performing the same metabolic process, how are they occupying exactly the same environments? And for this, we need to again go back to Darwin. So shown here, we have a sketch of Darwin's finches. Now, these are the Galapagos finches, and this is one of his sketches. And he noted, whilst in the Galapagos, that there is variation between the species of finch and the size of their beak, and how they foraged, with the size of their beak informing their foraging ability, with birds with larger beaks being able to more effectively forage and break up in nuts, and other size beaks being able to forage for other different resources. This is their niche, their ecological niches. So they're exploiting different resources. So whilst they are similar in nature, they can occupy the same geographical space because they occupy a different ecological one. They're adapted to a specific environmental set of parameters. If we go back to the sulfur oxidizers, whilst sulfide plus oxygen will potentially result in the formation of sulfate and the generation of energy within the cell, for each metabolism, there are multiple different pathways. And on the right here, we have a representation of sulfide oxidation of just two of the different potential pathways. We can see in pink, there is one pathway, and in purple, there is another pathway. However, we can also see in pink, some microbes have this enzyme, sulfite dehydrogenase, which provides a cheat. They don't need these two enzymes because they're producing this one, and it lets them skip a step. Each enzyme in these different pathways will have specific requirements, so metal ions that they need for their active sites. They'll have different affinities, so they will be able to bind to the sulfur species more effectively than others. And there's going to be different tolerances of pH and temperature, both within the proteins themselves and within the cells that house them. Therefore, whilst an overall metabolic reaction of sulfide plus nitrates or oxygen can give a theoretical yield of energy, there are many factors to consider. There is also the point that many organisms are incredibly versatile. This isn't completely true in terms of carbon source for the sulfur oxidizers, which tend to be autotrophic, but it is true in terms of the electron donors and acceptors they can use. For example, many sulfur oxidizing bacteria, such as the example here of a Campyllo, which was isolated before the reclassification, which can use both sulfur and sulfide as an electron donor and oxygen and nitrate as an electron acceptor. And many members of Campyllo, Gamma, and Aqua can also use hydrogen as an electron donor. So it's, again, not as simple as this one pathway. Many organisms are able to exploit several. So how does this relate to the microbes in the vents, and how does it relate to Darwin's finches? Well, in the same way that these finches occupy different ecological niches, so too do the three different phyla of sulfur oxidizing bacteria. Aqua tends to occupy a very narrow niche because it's more thermophilic than the other members of the community, but less competitively efficient at lower temperatures. So it occupies a band around here. Campylobacteria typically dominate the hydrothermal vent system below the aqua in proximity, but dominating numerically. And this is because they have a very high tolerance to sulfide, meaning that they can grow in close proximity to the mouth of the vent. Whereas the gamma proteobacteria have a lower tolerance of sulfide, but a much higher tolerance of oxygen, which means with reduced proximity to the vent, they're able to grow where the temperature has decreased, the sulfide is lower, but more oxygen is present. So they are all occupying a different niche within this selective site. And what if we change the geology of the chimney? So here we have the community in a mafic chimney. And if we change it to ultramafic, and we change the chemistry of the fluid from being both sulfide enriched to also hydrogen and methane enriched, instead of methanogenic archaea, we now see methanotrophic archaea, which we'll be discussing soon, as well as hydrogen oxidizing bacteria, in addition to the sulfur oxidizers. So methane oxidation, one of the areas I worked on for my PhD, so I to this day still find it fascinating, has an intriguing differentiation between its taxonomic distribution. What do I mean by this? Well, methane can be oxidized as a carbon or energy source by both bacteria or archaea, but it's obligately aerobic when done by bacteria, which some representatives found in hydrothermal vents are gamma proteobacteria, but there are other bacteria that can do this. But they require oxygen for this metabolic process. This is the pathway shown here. Whereas archaea that perform methanotrophy, the consuming of nitrate, are obligately anaerobic in doing this. And they can use sulfates, nitrates, or iron as potential electron acceptors. So as you can see, it's an incredibly versatile metabolism that allows them to exploit the methane and the other abundant species present within the system. Then there is hydrogen oxidation. Along with methanotrophy, this is one of the most prolific energy-yielding metabolisms in ultramafic systems because the geology varied, therefore so did the fluid chemistry, and microbes are exploiting the available electron donors. Potential electron acceptors for this metabolism are also incredibly diverse. So there's metals, sulfur, sulfate, iron, oxygen, nitrates. So again, and we see that Campyllo, Gamma, and Aqua are also groups that contain microbes that are capable of this metabolic reaction. There is also the delta proteobacteria making their first and only appearance in this talk, as well as hydrogen oxidizing archaea, which occupy a unique niche within the system because they have a very high affinity for hydrogen, which means that they can occupy further away from the vent and still exploiting the reduced but still enriched concentrations of hydrogen. But are there other types of vents other than just the active chimneys within the deep sea system? Well, one example and answer in the affirmative to this are the shallow water hydrothermal vents, which are heated by tectonic activity near plate boundaries. And you're a shallow water vent if you occur at less than 200 meters. There are 70 that have been identified, and of these, over half are associated with volcanic activity. We can see from the picture here that they are so close to the surface, you can in fact see the bubbling at the level of the ocean, at the surface of the ocean. So sulfur oxidizing bacteria are still highly abundant in this environment, and we still see archaea as highly abundant surrounding the mouths of the vent as they're so thermophilic. However, there is a much higher abundance of phototrophs and heterotrophs. As we can see very clearly, these vents are present within the photic zone. Light can reach here easily. So what are the specific differences? Well, the pressure is lower, meaning there isn't a requirement for piezophilic adaptations. But the environment is more impacted by physical processes such as storms and waves, which will have an impact on the microbial community also by inputting material and potential nutrients of another source. The waters are often less saline, and as previously mentioned, as the vents are within the photic zone, they are more productive. Chemosynthesis and photosynthesis co-occurring, which means there's more organic carbon, which means those microbes that require organic carbon are able to thrive. In similarities, the microbial community is still controlled by the temperature and the nutrient availability, which is again controlled by the geology, with the mixing waters also promoting the formation of sulfur-rich precipitates. So in this way, the microbial community stays broadly similar. Vents can also go extinct. And when this happens, there is a decrease in temperature, there's a decrease in the supply of reduced chemical species, and there is an accompanying major shift in the microbial community. Now, it's problematic to identify how this exact succession of one community to another happens because of requiring having analyzed the vents prior to going extinct and then analyzing the vent shortly after going extinct. So it shifts from this fluid actively coming out to having just the chemistry of the chimney itself. And we see that the microbial community shifts from fluid controlled to mineral controlled. As these precipitates have been forming, they have been leaving behind a legacy of abundant nutrients. With the reduction in sulfide concentration, the gammas are now able to outcompete and effectively exclude the campylobacteria, leaving them as dominating the process of sulfur oxidation. But sulfur oxidation is itself displaced by iron oxidation, which now plays a much larger role within the vent system and becomes the dominant metabolic process. This is performed by zeta proteobacteria and gamma proteobacteria of a different type. And talking about having to analyze vents shortly after they've died, there is this fantastic paper by Hu et al. In 2020, where they were able to analyze a hydrothermal vent and then another nearby hydrothermal vent that had only gone inactive 7 years previously. So this is the closest to identifying the active community transition from one point to the other. So iron oxidation. In extinct vents, iron oxidation is the dominant metabolic process. It's performed by either heterotrophic gamma proteobacteria or autotrophic zeta proteobacteria. So again, there is an intriguing difference between the taxonomic distribution of this metabolic process. And once again, it's a metabolic process that can occur in the presence of either oxygen or nitrate, with nitrate-dependent iron oxidation having been considered as another potentially Mars-relevant metabolism, with a review paper on this having been produced by my colleague in the group, Dr. Alex Price. There is also an abundant source of oxidized metals within the system, so iron, but as well as this, there's the manganese, copper, and the lead, which can be used as potential electron donors for both bacteria and archaea. So as well as the iron oxidation, there can also be iron reduction ongoing, so exploiting the chemistry that has been left in the vents. So we see here how this change in selective regime results in the loss of the ecological niches of many of these microbes, with a change in the chemistry resulting in a major shift of the species. A great example of this being the campylo, which lose their ecological niche of being able to tolerate high sulfide concentrations. When the sulfide concentration decreases, they are competitively inferior to the sulfur oxidizing gammas. There are, of course, in addition to the chimneys, other environments that are enriched by the geologically hot fluids in a very similar manner, and that's specifically the subsea floor. The water isn't just hot on the way out, it's heated as it goes towards the chimney. So let's take a second to focus on the subsea floor microbial biosphere and how this varies. So the mixing with the ocean water occurs below the sea floor, and this fuels a microbial community that's incredibly similar to that of the chimneys, with the same distance from the vents. So it requires the water to be sufficiently enriched in fluids, but it needs also there to be the habitability zone of below a specific temperature. And these microbial communities are also dominated by the gamma, campylo, and aqua of the sulfur oxidizers, as well as the methanogens. So it's effectively just from the other side of the water going in and out that we see this similar microbial community being enriched, which makes sense because, of course, it's the same geology. And if we're considering the water going in and the structure that is formed as the water is ejected, there is one further environment that we need to discuss when considering the environments within hydrothermal vents, and that is the plumes themselves. So the enriched waters of hydrothermal vents can create plumes, and these rise hundreds of meters off the seafloor. And whilst diluted with increasing distance, they are still enriched in many chemical species relative to the seawater itself. So these plumes can support microbial growth, and this then can then have a major impact over a much broader scale of the ocean's microbial communities and subsequently biogeochemical cycling. If we look at the microbial community that occupies the plumes relative to what's present in the subsea floor and the chimneys, there are occasionally shared members. But if we again think back to the concept of an ecological niche, these microbes on the chimneys and in the subsea floor are adapted to the higher temperatures and the more concentrated fluid chemistries, meaning that in the more dilute plume environments, they're in fact at a selective disadvantage, so do not dominate. And different sections of the plume act in very different ways. So in the rising plume, you have microbes that are resident only for a few hours due to the flow of the plume itself, with a few hours being a very significant amount of time for some microbial species, especially if they were previously nutrient deprived. Whereas the neutral plume, these can be occupied for much longer timescales and have something approaching a stable microbial community. So if they're not colonized by the chimney community or the subsea floor community, who are they dominated by? And they're in fact dominated by the microbes that are in the oceanic waters surrounding, who can gain access to this increased nutrient concentration, allowing increased growth over transient periods. Then, as we see over great distance, if we consider if there is a continual dilution, we see gradation in niches across the plumes as distance increases from the hydrothermal vents. And as we consider that these cells will be growing and then potentially being moved away by the actions of the plume or by oceanic currents, this impact with the transfer of nutrients and the transfer of cells which have increased in abundance can extend for miles away from the original vents, which has therefore a major impact on the general ocean community, and shows that when we consider the habitability of an environment and the impact of the microbes on that surrounding environment, we need to consider at multiple scales, both the hydrothermal vents and at both a broader and micro scale. But then the question is, if the water going in that produces the hydrothermal venting, the water that comes out to the vents, and the structures formed by the water itself create these multiple environments that are exploited and colonized by multiple different metabolic strategies, is there another environment for microbes within the vents themselves? And it leads on to the introduction of course, another major occupier of the system surrounding hydrothermal vents, and that is the animals. So I will be using the generic names for all of them, apart from where the actual species name is relevant, but there are some animals that are incredibly abundant in hydrothermal vents because they are incredibly productive systems, and this productivity doesn't just benefit the microbes and single-celled life. So we have the tube worms, we have crabs, we have mussels, snails, and many other different species. This is also my favorite picture of a crab for reasons I'll be going into. So there is a rich diversity of animals that survive around hydrothermal vents, but of course in the cooler waters. There are some animals that can survive incredible temperatures, but within the hydrothermal vents, they typically occupy those below 55 degrees centigrade. And there are many animals that are what is known as endemic to hydrothermal vents. So they are exclusive to hydrothermal vents, with some species being exclusive to an individual vent system. And there is an additional strategy employed by some of these animals occupying the hydrothermal vents beyond simply taking advantage of the higher levels of carbon and subsequent productivity. So they require a way of acquiring carbon and energy. Now, for many animals, this means simply that they eat either the bacterial biofilms that form along the chimneys or around the rocks surrounding the hydrothermal vent, or the phytoplankton, which are themselves growing on the bacterial cells. And it's worth noting that animals cannot perform sulfur oxidation or carbon dioxide fixation. So these two productive pathways, they cannot access. Another incredibly relevant factor for their survival, in addition to accessing carbon and energy and being in the cooler waters, is the dilution of metals and the other specific reduced chemical species. So sulfide is something we used routinely in the lab, and we have to be incredibly careful. And of course, the other metals available for the process of metal reduction include lead, copper, iron. Once again, there are no safe chemical species, there are just safe concentrations. And surrounding the vent, this is vastly exceeded for many of these chemical species. So what many of these animals do is they try to export the metals as quickly as possible, or localize them within specific subunits of their body, which prevents the damage from being excessive. But what many animals also do is they have a symbiotic association in order to enhance their survival. So when we're talking about a symbiotic association, we're talking about a mutualistic relationship where both partners benefit, in this case, the animal and the microbial symbiont that they have formed a relationship with. There is also commensalism, whereby some microbes benefit, or one partner benefits and the other doesn't. And if we go back to this picture of a crab, we can see this fur-like substance growing on it. This isn't fur, this is a microbial biofilm. And these crabs have been researched, and whilst there's evidence they have symbionts, it's within them. It's not this external sheath surrounding them, meaning that most likely these microbes are simply using the crab for transportation and access to different portions of fluid. But there's no reason to believe that the crab is being harmed. Were it being harmed, or if we think back to the pathogenic strain of E. coli at the start of this talk, this would be an example of parasitism, where one benefits and the other is suffering a negative impact on its either evolutionary fitness or health, such as many disease-causing organisms could be classified as. And what's incredibly exciting about symbiosis within hydrothermal vents is they have arisen multiple times in multiple different ways. So shown here, we have some pictures of Rimicaris, the blind shrimp, which are often very abundant within hydrothermal vent systems. Now, these have what's known as ectosymbionts. So they grow on the branchial chamber of the shrimp, and they are external, they are not internalized. There are some gamma proteobacteria and campylobacteria. And I haven't used the colors here because whilst it's possible that they're sulfur oxidizers, the exact metabolic relationship that the microbes are performing is unknown. Then there are, of course, endosymbionts, so internalized microbial symbionts. So we have the tube worms, the mussels, and we have species of snail. And these all have sulfur oxidizing gamma symbiotic relationships. So animals possessing symbionts that host them internally typically have to have specialized organs or tissues. And even in the ectosymbiotic relationship that the shrimp have, they still have enlarged branchial chambers to host these microbes. In the case of the mussels, they house their symbionts in their gills. Whereas in the case of these snails, they house their symbionts in an enlarged esophagus. So this is unusually enlarged in these species in order to better house the microbial cells. And this changes the nature of the symbiosis because being in the gills, the symbionts of the mussels have better access to the external environment and therefore the reduced fluid chemistry. Whereas the snails, it's in their esophagus. So the snail itself is supplying the symbionts with this reduced fluid chemistry. And it's also worth noting symbiosis can be with a community or a sole species. So chrysomalon snails have a sulfur oxidizing bacterial symbionts in the form of a gamma proteobacteria. Whereas some species of mussel that occur in hydrothermal vents have both a sulfur oxidizer and a methane oxidizing gamma proteobacteria, working together as well in symbiosis to supply the host. Whereas some species of mussel, in fact, just have the one. And these symbioses have arisen multiple times independently. So this is where the Latin names of the snails become relevant. So we have Chrysomelon, as previously mentioned, and we have Gigantopelta. These both have esophageal symbionts. They're both in the same family of snail, and they both have these sulfur oxidizers within them. So if we look at the family tree of these snails within the hydrothermal vents, they are not that closely related. And it's not true that they evolved from a common ancestor that already had this symbiotic relationship developed. In fact, they have many sibling taxa within the hydrothermal vents that survive without a symbiotic association, meaning that these two lineages of snail, in fact, separately evolved a symbiotic relationship with a sulfur oxidizing genus of bacteria. And Chrysomelon only has the one, whereas Gigantopelta also has a methane oxidizing gamma proteobacteria within its esophagus. So the symbiont is supported by the host. This is why, if you think about the evolutionary success of this strategy, they could be considered in some ways to be more successful than their wild living relatives. They have a more stable supply of specific nutrients, and they're provided with a stable environment. Stable italicized because, as we've seen from the chimneys going extinct, stable in geological terms doesn't truly exist in terms of environment and niche that microbes can occupy. But from the point of view of an individual generation of microbe, the tube worms are an ideal environment. So tube worms are a fascinating example of symbiosis because they also house their symbiont within a specialized organ. So if we look here, we have this coelom within the tube that houses the trophosome cells, which is chock full of these blue bacteria, and it's right next to capillaries. So what happens is, in addition to housing them within these specific organs, they produce a version of hemoglobin like we have. But in addition to binding oxygen, it binds sulfide. So being internalized, the tube worm has to supply them with these nutrients. So it binds to their version of hemoglobin. It then goes to the trophosome cells, where the microbe performs sulfide oxidation and carbon dioxide fixation, which then supplies the cell with some of these carbon compounds. So we can see that in exchange for supplying it with this safe environment and some specific nutrients, the host is itself supported with carbon and chemical energy from the symbiont, as well as detoxification of metals. Microbes are much more efficient at converting these harmful metal ions into less readily soluble and therefore entering into them forms. Many endemic event animals do not have these symbiotic associations, and they are able to simply thrive on a more productive system. But those that do have these symbiotic relationships have access to energy and carbon resources that their non-symbiotic sibling taxa do not have. But many are then dependent on the symbionts for survival. So there are many organisms that have either reduced or missing digestive systems. And this is true in the tube worms, and it's true in the snail species that we discussed that have reduced digestive tracts. In the case of the microbe, intriguingly, many of them are actually capable of surviving external to the host. And simply in the case of the muscle symbionts, upon the death of the muscle being released and then waiting for naive, newly born muscles to arrive in the environment, allowing them to colonize them. But in many cases, symbiotic bacteria do eventually also become dependent on the host, with the host being the selective environment in which they survive and not the hydrothermal vent, meaning that the selective regime in which they are surviving promotes a reduction in genomes and a loss of some features, because why would they need to produce all of their essential cofactors and vitamins if the host is supplying it with them, eventually resulting in a complete codependence. So we've discussed the different environments within hydrothermal vents, the viabilities of different metabolisms, and the different strategies and success therein. And we need to consider scale and biogeography when we discuss the evolutionary and ecological success of different strategies in different environments. Now, we've briefly touched on, is there migration of species between the different sites within a hydrothermal vent? So the chimney, the subsea floor, the plumes, and the animals. Well, we see that it depends on the level of taxonomic classification you look at, which is why here you need to go to lower levels. we see that members of the same family and genera are detected between the different environments. The animal symbionts that are sulfur oxidizing bacteria are relatives of the wild free-living sulfur oxidizing bacteria. However, if you go down to the species or strain level, it's the case that it's them being related and not an example of migration. So a gamma proteobacteria living on a chimney and a gamma living inside a tube worm are related. It's not a case of the free-living one easily being able to just colonize the animal. And this lack of migration relates to the concept of niches. We also touched on this with the plumes not being dominated by the chimney and subsea floor microbes. But what about within the same field? What if we have two chimneys within the same field? Well, within a field, there is variation in geology. And furthermore, at the micro scale, there is significant variation in toxins and resources between the chimneys. So whilst if these are both mafic systems, the fluids will be dominated by hydrogen sulfide, and therefore these will be colonized by sulfur oxidizing bacteria, there will be variation in the relative regimes between the two. If there's a slightly less flow of sulfide in one relative to the other, then gamma will be able to succeed. If there's a greater preservation of heat, then aqua will be able to better colonize in these environments. So at the functional level, we will still see that there is some conservation of function, but there may be this continued variation in taxa, even within the same field. And how do vents vary between sites? Well, different areas tend to have different species with many endemic taxa. This is true of animals, but this is also true of some microbes, to the extent that species may exist within microbes. Now, this is especially true between geologically distinct vents, so ultramafic, mafic, extinct, and shallow. But it also persists in geologically similar systems if we think of just two mafic vents, because again, we need to think at the scale of the microbe. At the micro scale, there will be sufficient variation, but there is also the constant issue in microbial ecology of we need to also try and separate dispersal from evolutionary success as well. It's possible that these communities are different from each other in taxonomic makeup, not because of selection promoting one group over another, but simply because one group didn't reach that site and therefore hasn't been competitively excluded because it's never been there. And many of these endemic taxa also arise due to an incredibly important evolutionary concept known as speciation, specifically allopatric speciation, which we're going to walk through now. So if you have the original population, and let's imagine that this is microbial cells of thermophiles that are being moved through the ocean on a current, we will then have possible events happening that will geographically separate a part of this population from the rest. So maybe some fall from the current and come to colonize a hydrothermal vent. Over multiple generations, and again, we need to consider that microbial generations are much shorter than for animals and plants, there will be an occurrence of mutations in one line different to the other. There will be different selective regimes. There will be different random selection and changes in the population's genetic makeup between the two, just because of random change and variation, as well as population bottlenecks. And this will eventually result in a genetically distinct species arising in one vent relative to the other. Although this is slightly more controversial with microbes because sex and sexual separation between microbes is much less of a thing than it is in animals. This is still how you end up with endemic microbial taxa. And it also doesn't have to be colonizing a hydrothermal vent. This could be microbes already within a hydrothermal vent over millennia developing a symbiotic relationship with an animal host. Again, there will be a different occurrence of mutations, a completely different selective regimes, and selection for a genome reduction, with also access to less DNA from other microbes, meaning that it will undergo a drastic change, eventually resulting in a symbiotic taxa and a free-living taxa to which it is related. And these also don't have to be microbial cells. This could be shrimp going on the ocean currents and then colonizing a hydrothermal vent, and again over millennia ending up being evolutionary distinct but related to other shrimp species. So we've discussed different strategies by which organisms are capable of surviving under the extreme selective regime of hydrothermal vents. We've discussed different metabolisms, different adaptive strategies that extremophiles employ, and the potential role of symbiosis and cross-feeding on available carbon. And just to end the talk, we're going to touch on mobile genetic elements, what they are, and the proposed role in enhancing survival in hydrothermal vents. So hydrothermal vents harbour a large diversity of what's known as mobile genetic elements. So these are, as the name would suggest, packages of DNA or RNA which can be transmitted between cells. So to use a metaphor, here we have a cell, and the bacterial DNA is the genome. So this is the standard hardware that you would have on your phone, and the plasmids are the apps, effectively. This means that they are transmissible, they vary in size, and they vary in utility and function. Now, if we consider that we've established that communication is very important, transport and concentration of metal cells outside of the cell, being able to deal with toxins, and being able to perform metabolic activities that give you a selective advantage within this incredibly competitive environment, some of these plasmids may contain the HSL coding genes, the communication molecule encoding genes, and some of them may encode a gene that allows the cell to fix nitrogen or better tolerate higher concentrations of lead. And these can be transmitted between cells because the plasmid is replicated. This is also a very common way through which cells acquire antibiotic resistance, because the plasmids are transmissible. And keeping with the metaphor of the phone, there is, of course, another way that data can be downloaded to your phone, and that is through viruses. Now, shown here on the right, we have what is known as bacteriophage. And these are viruses that specifically infect bacterial cells. There are also phage for archaea. We have these different structures here. So the DNA is contained here within the head, and then this is the tail, and it injects its DNA in, and it takes over the machinery of the cell, much in the same way a computer virus takes over a computer. So how this works is the virus attaches. This is the genome of the microbe, and this is the genome of the virus. It inserts its DNA into the genome, and it then has parts of the DNA which make the cell actively express and replicate the DNA and express the genes producing proteins that make up this structure. So the cell produces these in abundance. It then assembles them into viruses, and this then causes the cell to burst open. Now, if we take a step back to the definition of life that we're using for this talk, the reason that viruses aren't considered as living organisms is that they are not self-sustaining. They require a host in order to replicate in the same way that the plasmids do. However, they are capable of Darwinian evolution because they have DNA or RNA which undergo mutation and transmission between generations. But how does bursting open these cells have an impact on survival strategy? Why are they being considered in the same realm as the plasmids? Well, it's because they aren't only performing the lytic cycle. Viruses can also perform what's known as the lysogenic cycle. So the first step is the same. The DNA is injected and it inserts it into the genome of the microbe. This then replicates, and every time the cell replicates, it's also replicating the virus. Now, a couple of different things can happen. The cell can be stressed, and this will then trigger the expression and ejection, effectively, of the virus, which will then burst open the cell. Very famously, there's a strain of E. coli which produces a lethal toxin. It itself does not possess the toxin. The toxin is encoded by the viral DNA in its genome, and when the virus ejects because it detects antibiotic treatment, this is when the toxin is produced. Another thing that can happen is, following rearrangement of the genome and replication, there can be errors which introduce a portion of the microbial genome into the virus itself, meaning that when it's going between host to host, it's transmitting these additional functional genes, the same as the plasmids. And lastly, it's possible for, following replication error and recombination of the DNA of the cell, for effectively the virus to end up trapped, becoming a portion of the genome forever, and no longer being able to activate the lytic cycle. So viruses are highly abundant in hydrothermal vents, and in one vent system that was studied, populations were shown to be stable over time, and 40% of the identified microbes contained a phage within their genomes. And this isn't something that's exclusive to microbes. 8% of the human genome is viral in origin, with some of the genes encoded by this viral DNA providing important roles in the development of humans in the embryo. So it's been proposed that the transfer of genes, known as horizontal gene transfer, either virally mediated or mediated by plasmids, is more common in vent environments based on the detection of their high abundance, and that this may potentially play a role in enhancing survival and driving evolution with the transmission of survival enhancing genes between different microbial lineages, independent of vertical transmission. So in conclusion, hydrothermal vents represent an extreme environment with an array of selection pressures, and they're dominated by extremophilic microbes, with active hydrothermal vents dominated by sulfur oxidation, ultramafic systems by methane and hydrogen oxidation, and extinct chimneys by iron oxidation, with of course these metabolic strategies and success varying over time and space. There are multiple different environments that are specific to hydrothermal vents that the microbes are able to colonize and thrive in, and this includes many of the endemic species of animal. And survival in hydrothermal vents has been proposed as being enhanced by the transfer of genes between microbes, either by viruses or through transmission of plasmids. Thank you very much. I think we have time for questions. I will stop share.
Indeed, thank you very much, Michael, on behalf of everyone here for a fascinating tour through the microbiology of hydrothermal systems. And you are indeed correct. We have a number of questions, so we'll attempt to work our way through as many of them as we can. So we have a question from Lorenzo Carre, who asks, what is the relative abundance or importance of I should start with the previous question. You say that sulfur oxidation drives primary production in aphotic environments. So how much does methanogenesis represent?
That's a really good question. So the reason that methanogens are able to thrive in the chimney environment is because that functional group contains many hyperthermophilic microbes. In terms of abundance and relative abundance, it will very much depend on the specific vent itself. So what I've done for this talk is very much generalize the metabolic processes that dominate. So sulfur oxidation dominates in mafic systems and in most hydrothermal vent systems. Methanogenesis is typically also present, but for the specific relative impact on primary production, it really would depend on the environment itself.
And following on from that, what is the relative abundance or importance of non-methanogenic and non-methanotrophic hyperthermophilic and autotrophic archaea in hydrothermal vent systems?
That's a very good question. So in the interest of crafting this talk, I did exclude one very important group of archaea. So they are the ammonia oxidizing archaea. So hydrothermal vent fluids are enriched in ammonia, and they are able to use that in the same way that sulfur oxidizing bacteria use reduced sulfur compounds. So these also play a role in the microbial communities of the evolving hydrothermal vents. What I did for the purpose of this talk was focus more on the most abundant. So they are present and they do have an impact on the nitrogen cycling within the hydrothermal vent system. The precise impact and abundance will again depend on the precise system because they are typically more dominant within, I believe it's more acidic pH environments, whereas in higher pH environments, it's ammonia oxidizing bacteria, which typically dominate. Okay, thank you. Next question is from Suryendu Saha, who asks, what kind of biosignatures are we looking for in the cosmos? As you mentioned, extreme is a relative term. What is your opinion for the definition of the term habitable? So that's two great questions. So thank you very much. So in terms of biosignatures, what we do in our group is we attempt to perform simulation experiments with the necessary abiotic controls in order to identify if there is an unambiguous shift in chemistry or formation of a specific mineral or class of minerals that occurs only in the biotic test group. And it's incredibly hard. So we need something that's unambiguous, and we need something that would not be able to form in the absence of biology, which is incredibly hard to narrow down. So we're still looking. We're focusing currently on secondary alteration minerals and whether there are specific chemistries that of precipitated minerals that occur solely in the presence of microbes. But that's because my research currently focuses on Mars and the Martian subsurface. I think you would be completely looking for something different in the case of if you're working for, say, on the icy moons. So Dr. Johnny Tan also has some really interesting work published on preservation of lipids, because this is something that's conserved across all life on Earth. As you mentioned, extreme is a relative term, opinion in defining habitable. Professor Charles Coquelle from the University of Edinburgh produced a really interesting paper on habitable is a binary concept. It's incredibly hard to define, because if you perform an experiment to test whether microbes can grow there, if they don't grow there, that doesn't mean that another type of microbe may not be able to grow. And that's something we find in our simulation experiments. Sometimes methanogens don't grow, sulfate reducers do, or sulfur oxidizers do. So it really depends on what you test. And we know that some microbes are only viable in a community-dependent context. They depend on other members. I like the statement, it's a binary concept. It's either habitable or it isn't. Something can live there or it can't. I think the subsequent getting into the nitty-gritty of how do you define whether something is definitively uninhabitable relates to the concept that you can't prove a negative. I think it's very murky, but I do enjoy that paper as reasserting that a thing can't be partially habitable. It has to be a qualitative label of it's either habitable or it wasn't. And it may have become uninhabitable and it may be uninhabited, but it either is or it isn't. Yeah.
Thank you very much. The next question comes from Anurag Aithal, who asks, since the chemistry of vents is defined by underlying interactions, the metal contents will change across vents. Is this correct? And across vents, do we see metalloenzymes of similar functions using different metal centers, or is that conserved?
So the specific metal concentrations will vary across vents. There will be some shared when you have similar geologies. The exact metal ions that are used in enzymes, and this is talking specifically of the enzymes with which I'm most familiar, it's typically conserved. But there is often some wiggle room. So for example, in the case of the lanthanide dependent enzymes, they can use 1 to 4 of the lightest lanthanides, but they couldn't, for example, use iron or calcium instead in their active sites. So yeah, this is more typically restricted on what ion will actually fit inside the active site of the protein in the development of the protein. So that restricts it in terms of how they're able to vary in assembly quite a lot.
Okay, next question more concerns the practical aspects of science in these environments, and this comes from Niven Krishnan Santosh, who asks, how are such microbes collected from these environments? How are they transported from the environment to the lab? And is it easy to grow and store them later on in the laboratory?
That's a really fun question. So it very much depends on the type of vent. So shallow hydrothermal vents, broadly, I would say would be simpler to sample in because it's just simply easier to access them. You could see in that slide, there was a picture of a scuba diver just going down and collecting samples. So access is simpler there. When these samples are collected, pressure from deep sea hydrothermal vents, pressure is something that has to be accounted for, much in the same way that microbes that are hyperthermophiles require high temperatures to keep growing. What you don't want to do is accidentally lyse a piezophile by bringing it from high pressure to low pressure very quickly. So what they do is they'll collect samples under controlled conditions that allow them to still be cultivated. Is it easy to grow or store them? It depends on the individual microbe as well. It depends is a very common answer in microbiology. If they're hyperpiezophilic, if they're hyperthermophilic, then they require incredibly precise and specific culturing conditions and equipment to do that, whereas some microbes in extreme environments can sometimes be slightly easier to cultivate. So I recently worked on a sulfide spring in the Canadian high Arctic, and whilst there were some incredibly hard to grow strains, there were also a couple of weeds that you just chucked them on salty yeast media and they grew quite happily. For example, our glycerostocks made from the liquage. So I believe they're grown up and then sampled later. It's also worth noting that for many of these hydrothermal vents, and for some of the different taxa that are described, we know they're present from sequencing studies. So it's around 1% of all microbes can be cultivated, and it's because we don't know their specific requirements. Maybe sometimes they require other members of the community to be able to grow. So for some of the taxa that are described as present in hydrothermal vents, there are no isolated representatives, simply because they haven't got there yet. So the 1% is often taken as we will never be able to cultivate the rest. I like the viewpoint that as sequencing expands and we know more about their microbes and their DNA, we'll be able to better improve our ability to cultivate them and work out their specific needs that we aren't filling when we just chuck them in a tube in a lab. But yeah, so for a lot of these, there are some isolated representatives, and for some there are still none, and it's just through DNA itself that we are able to know that they are there and active.
Thank you. I really hadn't considered most of those nuances myself. The next question comes from Krishnendu Ghosh, who asks, for Darwinism or neo-Darwinism to occur, epigenetical changes are necessary. Do these extremophiles show such epigenetical perspectives for differential perpetuative gene expression?
So I may not be able to answer this question very well. I'm more familiar with epigenetics in human or eukaryotic context. So epigenetic, just to badly explain, are change. So a genetic change would be a change in the DNA that's transmitted between generations, and epigenetic would be more of a modification that doesn't change the DNA itself, but changes the way that the DNA is potentially expressed. I genuinely do not know the role that epigenetics plays in affecting the survival of extremophiles. I apologize.
Okay, so the next question comes from Barbara Cavallazzi, who asks, could you tell us more about the antiquity of methane-based metabolisms associated with vents? Methanogens and methanotrophs, for example, could they coexist in the same micro niche in the vent subsurface?
That is a really good question. Thank you very much. And I feel like all I am doing in this Q&A session is name dropping papers, but what's really cool about the relationship between the two metabolisms is they share, it's called hydrotetramethanopterin H4-MPT, as it's written in papers, which prevents people having to say the longer chemical name, is one of the key compounds that's used and involved in the metabolism of formaldehyde and the processing of one carbon compound. So they share this common ancestry, which itself is very ancient. So, and then it's the subsequent enzymology has evolved differently. There are environments in which they do co-occur, and I have read a paper where it was possible to cultivate methanogen and a bacterial methanotroph. So they would let the methanogen produce methane, and then they would inject in a little bit of oxygen, and the methanotroph would oxidize the methane. And it would, the idea of could there be this carbon cycle where one feeds the other Absolutely. I mean, that's been observed in the lab. And as with most things we observe in the lab, presumably it happens freely in nature, and it's just taken us a long time to observe it. But there is a really good review on that by Ludmila Chistosadova, Professor Ludmila Chistosadova, on the origin of C1 metabolisms in evolutionary timescales.
Okay, and staying on the topic of deep time and evolutionary biology, we have a question from Krishnendu Ghosh, who asks, were there any changes in vent microbiome at the pre and post great oxygenation event time?
So I've read about the great oxidation events, and I don't know about it specifically in the context of hydrothermal vents, but combining what I know, I believe that the direct microbiome may not have changed greatly, because the waters themselves are what is making the environment anoxic. So it's possible that it will have reduced the niche for the anaerobes, as is true for all life on Earth. If you imagine that for millennia, there was this submarine that all the cells were on that was leaking water, and then with the great oxidation event, it just burst open. It massively reduced the environments in which anaerobes were able to survive and dominate. But in direct proximity to the mouths of the chimney, I would imagine that this was a safe haven, in the same way that deep sediments in estuarine environments provided a safe haven for anaerobic microbes in the face of this massive toxin that is oxygen.
Okay, and I will add also that the fossil record around this time is very small, so it doesn't add a great deal of information to that. Our next question is from Lorenzo Carey, who asks, so macroscopic life is abundant in aphotic environments, but do you think it could have emerged without the development of photosynthesis?
That is a really good question, and I genuinely do not know. I would hate to answer and give just completely a guess. I mean, there are instances where it's hard to say. Similar to cave ecosystems, the animals that we see that depend on chemosynthesis are specialized versions of animals that typically exist in photosynthesis fueled ecosystems. So it's always very hard to look at that and say, could this have arisen if they were just here? I think it's an interesting question, but I do not know what the answer would be.
Okay, so we have the next question comes from Dana Camila James Fayado, who asks, you have talked about hydrothermal vents related to felsic rocks. What kind of reactions would take place here, and how much do they differ from those related to mafic and ultramafic rocks?
We are rapidly approaching my outer limit of what I know about geology. From my reading, I know that in the felsic hydrothermal vent systems, they're typically more abundant in hydrogen and methane. So in that way, you would expect there to be similar ecological niches to the ultramafic systems, and also a greater impact of pH, which is something that we didn't really get a chance to delve much into for the purpose of this talk. So pH would also be a major limiter in what taxa would be able to colonize those, but there are typically representatives of most functional groups that can survive, at least within the greater extremes of an environment.
Excellent, thank you. So then the next question comes from Arpan Kumar Basak, who asks, are there any studies considering extremophilic or hyperextremophilic communities, because there are extremophilic fungi and they have the potential to interact with bacteria?
Yes, so there are a couple of nice review papers on total community diversity. So as sequencing technology has evolved over time, it's gone from being able to sequence just the barcode gene that lets us identify what microbes are present to sequencing total DNA. And this is giving us a much better idea of total community because, of course, it's not biased by just looking at one specific gene or barcode. There are extremophilic fungi and protists and yeasts. So microbial eukaryotes do tend to get the short end of the stick in terms of microbiology talks. And I did gloss over them here because I was focusing more on the microbes fueling the system. There is still a much greater diversity, and there are some yeasts that have been found associated with some of the endemic animals in the same way that we have yeasts and tiny eukaryotic parasites growing on us, which is a lovely thought. But yes, so there are extremophilic eukaryotes within these systems, but again, the most extreme of the extremophilic are still the single-celled organisms in direct proximity to the vent.
Excellent, thank you. And the next question comes from Krishnendu Ghosh, who asks, what kind of bacterial quorum sensing mechanisms are expected in hydrothermal vent environments?
So given the importance of biofilm formation in colonizing of the chimney, there is a strong selection for the presence of effective communication and the production of homoserolactones, so the communication molecules that allow quorum sensing, the communication between different cells. In terms of if there are different systems, I believe that they are, one, quite ancient, and two, fairly evolutionarily conserved. So whilst there would be selection for having the ability to produce an abundance of homoserolactones, and there would be some restricted to specific taxa, I think that the systems that we observe in other microbial life outside of the hydrothermal vents would still be fairly representative of the system within the vents.
Okay, and the next question in the chat comes from Jyoti, who asks, are hydrothermal vents restricted to extremophilic microbial communities, or are there also extremotolerant microbes present? And if so, how do the extremotolerant organisms adapt themselves, or will they be mostly outcompeted by extremophilic microbes?
This is a great question, and it really does relate to the concept of the ecological niche. So I would say that all of the sulfur oxidizing bacteria that colonize the chimney are extremophiles, but if we think of how they have relative evolutionary fitness and are variable in how extreme they are. So the aqua are the most thermotolerant, which allows them to occupy this unique niche closer to the mouth of the chimney than the Campylobacteria. And then the gamma are less tolerant of the sulfide. In a similar way, there will be microbes at greater distance that are less extremophilic increasingly as you go away because there's less of a requirement. But it is the occupation, like if you're more tolerant to sulfide and metals and high temperatures, then you're going to outcompete something that is both struggling to compete against and deal with those pressures as well as competing with you for resources. But in terms of greater distance away, as we see with the plume community, there are examples of just extremotolerant or not even that extremotolerant with greater distance, where the extremophiles are suddenly also at a disadvantage. So success always depends on selection, which always depends on the environment. So there are extremotolerant in the broader hydrothermal vents and definitely in the plumes. It's also interesting whether we would consider if there was an obligate symbiotic species of microbe in an animal, as in it couldn't survive outside, even if it had extremophilic ancestors, we probably wouldn't consider that an extremophile anymore because it's no longer surviving in this actual extreme environment. It's adapted to the host and it wouldn't be able to survive in the extreme environment. So you can also, over evolutionary time, lose being extremophilic.
Splendid, thank you very much. I think we have 3 questions remaining, so we may yet make it through. The first of these comes from Anurup Mohanty, who asks, why do most extremophilic microbes fall under the domain Archaea?
So it's an interesting question, and it partially reflects the way in which archaea, I feel it partially reflects the way in which archaea were first discovered and then studied. So the first were identified in Yellowstone National Park, and these were the hyperthermophiles, and the most thermophilic organisms are in fact archaea. There's also, if we look at halophiles, there are halophilic bacteria and archaea. Halophilic archaea are able to tolerate much higher concentrations of salt, but the second this is slightly lower or temperatures are lower, the halophilic bacteria are able to compete more effectively, and there are in fact thermophilic bacteria as well. So these are traits which occur across the entire diversity of life. I think that it's potentially more of an artifact of the way that we have studied these organisms and the order in which we've discovered them. So, for example, going back to the point of we used to think all archaea were extremophilic, and there are definitely a lot that are, but if we were to sequence a soil profile or the microbiome of the leaf of a plant, there would also be abundant archaea, because there are many that are also what we would consider normal or mesophilic. So whilst there are many, there are also as many, or maybe not as many, it's hard to quantify these things, but there are also many extremophilic bacteria, eukaryotic organisms. And I can't be certain, but I do think that's potentially an artifact of how we've studied the organisms, which is, again, always a very important principle to consider when we think about ecological and evolutionary principles.
Okay, fantastic. The next question comes from Arunava Podar, who asks, how do microbial communities differ between hydrothermal vents and hot springs? What is your take on the origins of life theories in hydrothermal vent systems versus those in hot springs? I know this is a hot topic of conversation at the moment.
Yes, well, I have been heckled in the past for saying viruses weren't alive, so I'm quite enjoying the chat function for these discussions. Hydrothermal vents and hot springs. Well, hot springs are in the photic zone, so there is a much greater role of photosynthesis relative to the photosynthesis that's enabled by the glow of the hydrothermal vents, and then accordingly they're much more productive. In terms of origin of life theories, that's definitely not my specialty. I enjoy reading around it as the same way I enjoy reading around science fiction, because I think it's a fascinating field of study. There was a recent review paper in the journal Life that was done by a lot of early career researchers. The name escapes me, but it was a really nice review of the field, and it was talking about just the privileged molecule approach of how we consider specific molecules. This one will have come before the other, and how that's linked very much to, or if you think DNA came first or RNA, then you have to think that this environment is where it happened. I do not know, because my opinion is completely uninformed. I've read, but I don't want to say with any fake air of authority, because I've merely been reading as someone interested in the field as opposed to an expert.
Thank you very much, Michael. And the final question we have comes from Anurup Mohanty, who asks, when we say archaea are closely related to eukaryotes, does that mean after billions of years they will become more specialized or multicellular, or are these things more random?
That's a cool question, and I nearly included these slides. So eukaryotes are descended from archaea. So there were a few key steps that happened. One, there was the invagination of the membrane, increasing internal surface area to volume ratio, which is how eukaryotic cells got larger and also captured the DNA within a membrane. There was also then a symbiotic association developed with a proteobacteria, which over long, like evolutionary timescales, became an endosymbiont, resulting in the reduction of the genome of the proteobacteria to the extent that it became the mitochondria we see in cells today. And in plants, it was the same thing with a cyanobacterium. And the order in which those events happen is incredibly controversial. So I said them because that's the order that I happen to have them on a slide, but the order in which that happened is unknown. But it's always important to remember in terms of evolutionary questions is that selection is, of course, not random. Mutation is. Evolution itself doesn't have a direction. So this is why I made the point of there are no higher evolved organisms or more evolved organisms. Like if you compare me to a hyperthermophile living in a hydrothermal vent, if we swapped places, we'd both die. His success depends on the environment in which this has occurred. So it's hypothesized that multicellularity occurred as a result of either a DNA mutation that resulted in a successful strategy accidentally, and there's some fun studies on yeast where they purposefully introduce mutations. And there's also examples of multi-celled assemblages that increase survival, biofilms being one of these, and cable bacteria being another really exciting example. So there are ways in which working as a collective does enable success within an environment. But this doesn't mean that a billion years from now, that there will be another lineage of multi-celled organisms, because for that to arise, there would have to be a specific sequence of mutations and selections and survival. Random chance and drift plays a major role. For example, if that was to occur at the base of a volcano in a small pool, and then the volcano erupted, well, then it would all die off. So it's not so much that all things are random, it's that while selection has a direction in that, like, you either survive or you don't, evolution itself doesn't have a direction. It's not striving to be multi-celled. It just happens that the multi-celled organisms that evolved had a successful strategy.
Wonderful. Thank you. Thank you very much again, Michael. We have made it through all of the questions, and so I thank you again on behalf of everyone here for your time and your expertise this morning. And indeed, I think we were a virtual audience of almost 200 people. So that's very good to see. Thank you very much again, Michael. And I hope that everyone will join us again tomorrow when we will have a lecture seminar delivered by Dr. Francis Westall on hydrothermal activity and early terrestrial life. So we will see you same time, same place, cyberspace, for more seminar lectures tomorrow as part of the EANA Hydrothermal Spring School. Thank you again, Michael. Thank you, everyone, and have a good day.